Hi everyone, welcome to our module on normal hematological structure and function. Before beginning with the video, I'd like to request all of you to subscribe and keep supporting. This lecture series is provided free and is open access for everybody. If you find it useful and wish to support its development, you can pay with the link that is on the screen and also pinned in the description. You can pay with any of these payment methods. All right, in this video, we'll be starting with the absolute foundation for hematology, the normal structure and function. Before we jump into diseases like anemia or leukemia, you need to clearly understand where all these blood cells come from and how they're actually regulated and how they function at a molecular level. So if you understand all of these things properly, everything else in hematology will definitely become very much easier. By the end of this lecture, you should be able to explain how erythropoietic stem cells work, how blood cell production is regulated, and specifically how red blood cells develop. We'll also go into hemoglobin structure, oxygen transport physiology, oxyhemoglobin dissociation curve, and iron metabolism, especially hepcidin regulation, which is very high yield for all the kinds of exams out there. So think of this lecture as building your conceptual base for all the hematology questions. All the blood cells in the body originate from a single type of cell called as the pluripotent hematopoietic stem cell. These cells are located primarily in the bone marrow in adults. A very key important exam point is that the marker CD34 plus. CD34 positivity is a key feature for all the kinds of these stem cells, and this is how we identify stem cells in flow cytometry and stem cell transplantation. Functionally, these cells have two key abilities. The first one is self-renewal, meaning they can keep producing more and more stem cells from a single one. The second thing is multipotent differentiation, meaning that they can give rise to all the different types of blood cells from a single hematopoietic stem cell. So these are really helpful. So essentially, this one cell type is responsible for maintaining our entire blood system throughout life and to maintain lifelong hematopoiesis. Now, once the stem cell starts differentiating, it branches into two major pathways, the myeloid lineage and the lymphoid lineage. The common myeloid lineage gives rise to red blood cells, platelets, and most of the white blood cells like neutrophils, eosinophils, monocytes, and basophils, whereas the lymphoid lineage gives rise to B cells, T cells, and natural killer cells. This differentiation of myeloid progenitors and lymphoid progenitors is not random. It is tightly regulated by cytokines and growth factors in the environment of a bone marrow. So, depending on the signals the stem cell receives, it commits to a specific lineage, whether myeloid or lymphoid, depends on the signals that the stem cell receives. Hematopoiesis is not just about producing cells, it's about producing the right number of cells at the right time. So, this process is regulated by a complex network of cytokines, growth factors, and the bone marrow microenvironment. These signals control or determine three main things. The first one is cell survival, the other one is cell proliferation, and the third thing that these signals control includes the lineage commitment. So if this regulation is disrupted, it can lead to conditions like anemias, leukemias, or bone marrow failure. So understanding this regulation is critical not just for physiology, but also for pathology. Now let's focus specifically on red blood cell production, which we call as erythropoiesis. The key hormone here is erythropoietin or EPO. This is produced by the peritubular capillary interstitial cells that are present in the renal cortex. The main trigger for erythropoietin release is hypoxia. So whenever oxygen delivery is low, the kidney responds by increasing erythropoietin production. This is a very important physiological feedback mechanism. So whenever there's hypoxia, erythropoietin production is increased by the kidneys, and this leads to increased production of red blood cells. The regulation of erythropoietin is mediated by hypoxia-inducible factors, also called as HIFs. Under low oxygen conditions, HIF gets activated and acts as a transcription factor. So it increases the expression of the erythropoietin gene, which then increases red blood cell production. So clinically, this mechanism becomes important in conditions like chronic anemia, chronic hypoxia, or high altitude adaptation. Now let's look at how a red blood cell actually matures. It starts as a proerythroblast and then goes through multiple stages before becoming a mature erythrocyte. So during this process, three main things happen. The nucleus condenses, there's nuclear condensation happening, hemoglobin accumulates, and eventually the nucleus is completely extruded, nucleus completely disappears. So the different stages includes proerythroblast, basophilic erythroblast, polychromatic erythroblast, orthochromatic erythroblast, reticulocyte, and finally a mature erythrocyte. The key process that happen during this process includes nuclear condensation, hemoglobin accumulation, and nuclear extrusion. This results in a mature RBC, which is specifically optimized for oxygen transport. Before becoming a fully mature RBC, the cell enters a stage called as a reticulocyte. These cells, or these reticulocytes, contain some residual ribosomal RNA. So because of this, they appear slightly bluish on right GMs are staining, a phenomenon, and this phenomenon is called as polychromasia. Polychromasia. Clinically, reticulocyte count is very important because it tells us how active the bone marrow is producing, how active the bone marrow is in producing the new red blood cells. The main function of red blood cells is carried out by hemoglobin. Hemoglobin is a tetramer, meaning it has four globin chains and four heme chains. Each heme contains iron in a ferrous state, Fe plus 2 state, or a reduced form. And this Fe plus 2 form is essential for oxygen binding. This structure allows the hemoglobin to efficiently transport oxygen and carbon dioxide throughout the entire body. Now, a very important concept, hemoglobin can only bind oxygen when iron Fe plus 2 state or ferrous state. So if it gets oxidized to ferric state or Fe plus 3 state, it can no longer bind oxygen. So this condition where the heme is oxidized to Fe plus 3 state or ferric iron, and then this leads to inability to bind with oxygen. This is called as methemoglobinemia. Clinically, this leads to impaired oxygen delivery even if oxygen levels are normal. During development, hemoglobin composition changes. Fetal hemoglobin, or HbF, consists of two alpha and two gamma chains. HbF has a higher affinity for oxygen compared to adult hemoglobin. This is crucial because it allows the fetus to extract oxygen from the maternal blood across the placenta. After birth, there is transition from fetal hemoglobin to adult hemoglobin, which is HbA. HbA consists of two alpha and two beta chains. This transition typically occurs in the first 6 months of life. Clinical significance is that hemoglobinopathies often become clinically evident during this period or the first 6 months of life. Conditions like sickle cell disease, beta thalassemia, they become apparent only after the hemoglobin F levels decline. You should be familiar with different hemoglobin types and their clinical relevance. HbA is the predominant adult form. HbA2 is slightly increased in beta thalassemia minor. HbF is normally very low in adults. HB BARDS and HbH are seen in alpha thalassemia and are associated with severe diseases. These are very commonly tested in all the exams. The relationship between oxygen tension and hemoglobin saturation is represented by a sigmoidal curve. This shape is due to cooperative binding. When one oxygen molecule binds, it increases the affinity for the next one, and then it further increases the affinity for the next oxygen molecule, and eventually this continues as a cycle. This property allows efficient loading and unloading of the oxygen depending on how much oxygen the tissue needs. A right shift means decreased affinity of hemoglobin for oxygen. This is actually beneficial in tissues because it promotes oxygen release. Conditions that cause right shift includes increased H plus or hydrogen ions or decreased pH, increased CO2, increased temperature, and increased 2,3-bisphosphoglycerate. You'll commonly see this right shift in cases of exercise, hypoxia, and anemia, where there's more oxygen unloading at the tissues. A left shift is the opposite. It means increased affinity for oxygen. So hemoglobin binds onto the oxygen more tightly than before. The causes includes decreased O2, decreased temperature, fetal hemoglobin, decreased 2,3 bpg, and carbon monoxide. Clinically, this can lead to tissue hypoxia even when the oxygen saturation appears normal. Iron is essential for hemoglobin synthesis, but it must be tightly regulated because excess iron is toxic. Most iron in the body is found in hemoglobin. It is stored as ferritin and hemosiderin and is transported to the blood by a transport protein called as transferrin. An important concept is that the body does not have an active mechanism to excrete iron. The master regulator of iron metabolism is hepcidin, which is produced by the liver. Hepcidin controls iron levels by acting on ferroportin, the only known iron exporter. Ferroportin is present on the erythrocytes and macrophages. When hepcidin binds to ferroportin, it causes its internalization and degradation. As a result, iron gets trapped inside the cells and serum iron levels decreases. So this mechanism is central to many disease processes. I'll repeat it again. When hepcidin binds to ferroportin, it causes its internalization and degradation, and this prevents iron from being released into the bloodstream. As a result, iron gets trapped inside cells and serum iron levels gradually decrease. So, this mechanism is what we see in all the kinds of disease processes. In conditions like chronic inflammation, hepcidin levels increase. This increase is mediated by interleukin 6. As a result, iron gets trapped in the storage sites and is not available for red blood cell production. So, even though total body iron is adequate, it cannot be used in those cases of anemia of chronic disease. And that's the cause ultimately. It's like whenever there's chronic inflammation, the hepcidin levels also increase. So, this increase is mediated by interleukin 6. And as a result of all this, what happens at the end is that the iron gets trapped in storage sites and is not available for active red blood cell production. It is just stored in the form of storage proteins like ferritin and hemosiderin. It's not in the bloodstream, so it's not used for red blood cell production. So even though total body iron is adequate, free iron is low, so and it cannot be used. This eventually leads to anemia of chronic disease. Now let's quickly summarize the key points. Hematopoietic stem cells are CD34 positive. Erythropoietin is produced in the kidney in response to hypoxia. Fetal hemoglobin has higher oxygen affinity than adult hemoglobin. The oxygen dissociation curve shifts right with increased carbon dioxide, increased H+, increased temperature, and increased 2,3-bisphosphoglycerate. And finally, hepcidin regulates iron by degrading ferroportin. So if you understand these concepts clearly, you'll be able to solve almost most of the physiology questions related to the hematology system. I'll be back in the second video with a lecture on hemostasis and thrombosis. Before that, I would like to request all of you to subscribe and keep supporting. Thank you. Bye.